Hi, my name is Allison Romanski, and the co-researchers on this project were Madeline Lang and Dr. Peter Wampler. I go to Grand Valley State University, and my project this summer was the 2021 Grand River water quality sampling. This summer, we sampled 23 different samples of the Grand River from headwaters to mouth, and we did this to understand longitudinal variability in the river and to understand how different tributaries and different organizations along the river play a factor into the overall health and quality of the river. Our overall research goals are to sample different locations under different flow conditions, collaborate with wastewater treatment plants, and to develop a water quality index specific to the Grand River. Our methods for this project consisted of field sampling and lab analysis. For field sampling, we used a device called the In Situ Aquatrol 500 multi-parameter sonde for instantaneous field analysis. We also used a device called Wildco horizontal beta bottle to collect samples from the edges of bridges, and we used kayak to sample reaches that were not accessible by roads. In the lab, we used HACH analysis machines in order to analyze results such as chloride, ammonia, total phosphate, nitrate, and dissolved oxygen. We used a vacuum filtration system to analyze total suspended solids, and we used IDEXX quanti-tray 2000 to analyze E. coli and total coliform. We need both field sampling and lab analysis to collect all of the data for all of the parameters we needed to sample. As you can see here, we have preliminary results from our Saranac-Tololo reach that we completed on both June 3rd and August 16th. On June 3rd, which is the blue bars on these graphs, the flow rate was 774 CFS, and on August 16th, which is the orange bars on these graphs, the flow rate was 3,633 CFS. On August 16th, the flow rate was about 5 times higher, and that difference is shown through our data, especially in the E. coli and chloride graphs. The E. coli graphs show that on August 16th, the day of high flow, there was much higher levels of E. coli. This could be due to the heavy rain creating runoff, especially from agricultural areas. However, the chloride is shown the difference in the opposite direction. On June 3rd, the chloride was about twice as high as August 16th, and this could be due to the rainstorm diluting all of the chloride in the river. On August 4th and 5th, we took 36 samples of the entire Grand River from headwaters to mouth. The flow rates between these 2 days were not significantly different, so our results should be consistent. Sampling the river like this in such a short span allows us to see different trends that arise among our data. For example, by sampling this, we noticed that there was a high spike in the ammonia levels near the headwaters of the river. We used our data that we collected this past year and compared it back to old data sets that were taken in 1990 and 2020. Our comparisons have shown that there are a few trends that have occurred over the past 30 years. The most notable trends are among E. coli, dissolved oxygen, and temperature. The E. coli levels have decreased significantly over the past 30 years, where the temperature has increased about 4 degrees over the past year. Also, the dissolved oxygen has decreased by about 2 parts per million in the past year, which can affect plant life. The collaboration with the Allendale Wastewater Treatment Plant was both efficient and economical for both parties. We hope to collaborate with other treatment plants in the future. Also, the HACH method results are reproducible and compatible with results from duplicate samples sent to other labs. Additionally, the flow conditions, time of day, and order of sampling, upstream versus downstream, may influence the calculated water quality index values and sampling results. The entire river can be sampled in 2 full days if samples are collected using a tube sampler from easily accessible locations such as bridges. Sampling from a floating vehicle such as kayak may introduce sampling problems due to the water moving at the same speed to the boats. A significant increase in water temperature and chloride and a decrease in dissolved oxygen were documented for the 2021 samples for the entire Grand River. Special acknowledgements go out to the Allendale Wastewater Treatment Plant and their staff in assisting us in performing all of our tests and allowing us to use their labs. Also, acknowledgements go out to the Lower Grand River Organization of Watersheds in the city of Grand Rapids, who helped us run duplicate samples and organize sampling days with each other. Also, thank you to the NASA Space Grant and the Office of Undergraduate Research and Scholarships at Grand Valley. They both helped us with funding that made this project possible. I hope you enjoy my presentation. Thank you for listening.